Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Ghersi, and I'm a postdoc at Yale University. And I will be moderating today's session with Dr. Elle Roberson, a postdoc at the University of Texas. We are really excited to highlight the work of our outstanding postdoctoral members. Today, Dr. Alice Accorsi from Stowers Institute for Medical Research and Dr. Jiakun Chen from Vollum Institute, Oregon Health and Science University will share their research. Each speaker will give a 20-minute talk, followed by 10 minutes Q&A. Please enter your question in the Zoom Q&A box. It is now my pleasure to introduce Dr. Alice Accorsi, a postdoc in Dr. Alejandro Sanchez Alvarado's lab at Stowers Institute for Medical Research, where she studies adult regeneration and tissue plasticity. Alice is a brilliant cellular and developmental biologist. During her postdoc, she focused on establishing a new research organism that will impact the understanding of the visual system biology, evolution, development, and regeneration. Since Alice started her scientific career, multiple agencies recognize her work through multiple awards and fellowships, such as the American Association of Anatomy's Postdoctoral Fellowship and being selected as one of the leading edge 2021 fellows. Today, Alice will be presenting her current work on how Pomacea canaliculata represent a new model to study camera eye regeneration. Without further ado, I present Dr. Alice Accorsi.
Thank you so much for the introduction. Let me share. Is that, you guys can see it?
Yes, it is perfect.
Perfect. Thank you so much for the introduction, and thank you everybody for being here. I love that SDB is organizing this amazing seminar series, and I'm really glad to be here to share my work with you today. So as a scientist, I'm really interested in development and regeneration. And in particular, from the development, I'm really interested and fascinated how with a handful of cells, we can actually create every time a really perfect, organized and proportioned organism that then will grow as an adult. But we have to remember how many different tissue and different organs are creating this really complex structure. And to me, it's even more surprising and fascinating how during regeneration, we can actually rebuild individual components in an adult organism. So if you think during the development, all the cells are kind of knowing what's happening in the neighbor area. But if we have to regenerate, we have an adult organism that lost or damage a component of their body, and they actually can rebuild a completely new organ that is perfectly patterned like the old one, can match the size of the old organ, and will have to reconnect with the adult body. And so since I'm interested in these biological processes, I decided actually to explore these aspects utilizing eyes. And so from this slide, I hope you can appreciate how beautiful and diverse eyes can be in nature. But we have to remember that these eyes are also really, really important for us human. And here I'm showing you how our vision, it can impact by damage to the eyes, by aging or disease. And the main point is that we cannot regenerate our eyes. So if something happened to our precious retina or cornea, it's probably a damage that will last for all our life. And so this is bringing me to try to understand how eyes can be regenerated. And so for trying to understand and answer this question, that it's a really broad question, we can see which eyes are actually available to start trying to understand the process of regenerating the eyes. And here I'm telling you that eyes can be very different, and they are categorized based on their anatomical structure. And particularly human, they have the one that you can see here that we will call camera eyes for the rest of the talk. And so if we start focusing on the animals that we can have available in the lab to try to answer this question, we can have, for example, planaria with pigmented caps, where there is no chamber and no refracting device. That means that we have only like a thin layer of photoreceptors that can detect the light. More complex, more complicated are the compound eyes in many insects, where there are multiple chambers and refracting devices such as cornea and lens that can focus the light on the retina. And the camera eyes, if you remember, are the ones that we have also us as human beings, are shared among vertebrates. And so all of them, they have a single chamber and again, a cornea and a lens that is focusing the light on the retina. And if we ask, okay, if we want to study regeneration, which of these eyes can regenerate? Well, only the planaria can actually completely regenerate their eyes if we remove the entire structure. There are no evidence in literature for Drosophila or other insects to be able to regenerate their eyes if the entire organ is removed. And if we look more in detail on vertebrates, what we can find in literature is that there are some animals like salamanders or zebrafish that can regenerate some of the components, like a few cells in the retina or the cornea. But again, if we remove the entire eyeball, but they are not able to do a complete regeneration of this structure. And so here it's where me and Pomacea canaliculata, this freshwater snail that is an emerging research organism, are coming into the picture. So what I really wanted to do, it was trying to fill this gap where we needed an organism with a complex camera eyes, like vertebrates, but also able to completely regenerate if we were amputating the entire eyeball. And so I started like looking around on which animal could like match both characteristics, and I landed on this snail. So one of the first experiments that I did, it was obviously trying to ask if the eyes of these snails were camera eyes. And so you can see here the structure, the eye, you can always identify it because there is this black dot. And if we cut it and section, this is an hematoxylin and eosin section of the eyes of the snail, we can see if we move from left to right that there are many components that we probably are already familiar with. There is the cornea, that is the transparent epithelia that is letting the light reaching the lens, that is focusing the light on the retina, this layer of photoreceptors that are these specialized cells that can detect the light. And then these cells are connected to the brain through the optic nerve. And if we compare this structure to the human, here it is, it's actually really, really similar as a structure, like anatomically, to human and invertebrate eyes. Great, this is our first point. Second point, can these eyes regenerate if we do a complete amputation? Second experiment that I did, it was cutting the entire eyeball away. And so I made sure to cut at the base of the eye stalk to make sure that all the components that I just mentioned to you were actually removed. 6 days post-amputation, we can see that the wound healed and there is a beautiful blastema that it's reached of H3P and BRDU positive cells. So that means that there is active proliferation in the blastema. And a couple of weeks post the amputation, almost all the animals that we actually analyze, they are going through like a complete process of regeneration, and all the components that I just mentioned to you, the cornea, the lens, the retina, and the optic nerve, they are all again recognizable and visible. So this was a great win from my side, because now we know that we can use this freshwater snail to actually study camera eye regeneration. And so the gap that I was talking to you about, now it's actually like this animal can actually open the door to this new type of studies. And so far I showed you mainly like morphological analysis, but if we really want to understand how camera eyes fully regenerate, we need to switch more mechanistically and try to understand which like pathways and molecules might be really important for this process. And so for doing this, we need to bring these animals in the lab. And Luckily, these snails are really easy to maintain. They have a really abundant progeny. We were able to close the life cycle in laboratory condition, and then we also build a system where these snails are really happy and they can actually grow really fast, and they reach sexual maturity in 3 months. So their generation time is not that different from what we know already from zebrafish and mouse. I developed, in collaboration with another person in our lab, a protocol to do the karyotype. So we checked that they were actually diploid, that there was nothing like different going on this side. And I also participated in the effort to sequence and publish the genome for, again, for this species. So this was already like a really good starting point to try to get a genetic manipulation going. But for doing genetic manipulation, ideally, we want embryos. And so let me tell you a little bit more about where we can find embryos. These snails, they're not hermaphrodite. There are male and female that are separate. After breeding, this is a female. The female is crawling out of the water and is laying this round structure. They are 2 millimeters big. And you can see on the outside part, there is an eggshell that is going through calcification in the first 24 hours. What there is inside the structure? There is a yolk, that is the pink part, that represents the nutrients for the embryos, that is really small. It's 80 micron in diameter and is in the really middle of all this pink yolk. So first thing first, we needed to find a method to collect the embryos and look at them. So I developed an atlas with a stereo microscope, but also we collected a bunch of images at the LCM and with confocal microscopy, really to describe and understand the development of the snail. Here you have always the snail facing left on the anterior side, and the pink portion that you can see in the middle of the embryo is actually post-gastrulation, the embryonic gut that is full of this pink yolk that the embryo keep ingesting, and is full of nutrients that the embryo needs to keep growing. But something that I really want you to focus on is that by the time that this embryo is ready to hatch, is already looking like a small adult. This means that this mollusk, differently from many other invertebrates, is not going through a metamorphosis or a larval stage and a big change in their body structure, but they have a direct development. So all the structure that we can find in the adults, we can actually see them developing during the embryonic life. And mainly for, if you remember my introduction, my interest in development and regeneration, this is a really nice feature of this snail that will allow me to compare one-to-one features and characteristics that we find in the adult with the embryo development. And so I told you, now we can collect the embryos. We can, even better, we can collect zygotes. And the zygote is staying at the 1 cell stage for 6 hours before the first cell division. And this is a really important thing for giving us time to actually macroinject them. And so after testing a few different protocols, we found out that the best way to inject them is having a holding pipette, so we can keep the embryo still and we can inject these embryos. We try to inject as a first thing with exogenous mRNA. And so with this experiment, not only we tested that the embryos can survive the injection, but they can also use their exogenous mRNA. And you can see here that is a GFP fluorescent. They can use the exogenous mRNA, and this is actually opening up a full set of experiments for overexpression using this approach. But we didn't want to stop here, obviously. And so we kept moving forward and for being able to develop a maybe line in the future with mutant lines, we need to make these embryos grow as adults. And so for doing that, I told you that these embryos need the yolk extract as a nutrient because they don't have enough nutrients in the zygote to complete the development. And so this is a really important protocol that I also developed where I can collect and obtain a yolk extract, and I culture the embryos after manipulation, for example, injection. And here you can see how more than half of the embryos that we can culture in this way, like ex ovo culture, they can complete the development and they can grow as adults, and they will be perfectly fine, and they will breed and make a new generation of snails. So this was extremely important, not only for developing lines, but as you can see, now we can actually visualize the embryos. And so this is also opening up the possibility to do live imaging experiments. And so now that we have injection working, we can culture the embryos, we have all the tools required to actually try to see if we can create mutant lines. And for this, we needed a target. And maybe obvious choice was to try to target PAC6. PAC6 is a conserved transcription factor that is required for eye development in many species that has been studied. So we know that PAC6 is controlling the eye development in Planaria, in Drosophila, and in all the vertebrates that we know of. But what we don't know if it is also in this snail, PAC6 has been conserved with his role of developing eyes. And so this sounds like a really good target to test CRISPR-Cas9. So what we did, it was finding our PAC6 gene on the genome that we already published, and we designed guide RNA, we injected them, we bred the injected F0 with the wild type, heterozygous. It was already really exciting at this stage to see that we could have germline transmission. And then once we inbred the heterozygous, again, another really exciting moment where we can see that the allele was passed to the F2 generation with Mendelian inheritance rules, and there was nothing like too bizarre going on. And so with this, I'm showing you on this side of the screen, the wild type and the heterozygous. So we can clearly tell in these embryos that there is a well-developed eye stalk. The pigmentation is showing the eye with the retina that is forming and developing. And we can use the tentacle as a reference to find this structure. While in the mutants for PAC6, the homozygous animals, again, we can find the tentacles that is perfectly fine, but close to the tentacles, there is actually nothing. So not only the pigmentation is missing, but also the retina and the full eye stalk. So with this evidence, we can conclude that the role of PAC6 is conserved also in this snail for developing eyes. And so this is really interesting to find that not only there is an anatomical structure, but at least there is a PAC6, or there is some molecular similarities with the other animals that we know of. And so what is next? Now I'm set up in a really interesting moment where we have the tools and we have a really interesting biological question, and we can move on trying to understand mechanistically how the complete regeneration of complex eyes in adults can happen. And for doing this, we already have an intact eye single cell RNA-seq atlas. We found in this data set the photoreceptors markers, neurons, we validated with in situ, and it's going to be really interesting to see how this population change during the regeneration process. We want to define if and how the embryonic development programs are redeployed during adult regeneration. So it's something that I didn't specify you earlier, but during the embryonic eye development, we kind of see the same steps that we observe during eye regeneration, obviously post the formation of the blastema. And while we need 3 months to complete the development of the eyes, we actually need 3 months to have like a full, complete regeneration also of the eye. So the timing also match in a really interesting way. And we also already acquired a bulk RNA-seq during the development, and it's going to be a really important data set to start comparing the development of the eyes with the regeneration. And finally, today I talk to you only about a full amputation. So if you remember at the beginning, I told you that I cut at the really base of the eye stalk and remove all the components that are characteristic of the eyes. But what we can do is actually try to shed light on the role of the individual components during complex eye regeneration. And we can do that challenging the system with different amputation paradigm. For example, we can do lensectomy, so removing just the lens, or retinectomy, where we remove the lens and the retina, but without the clean cut of the full amputation, and see how the animals regenerate. I already can give you like some hints on something really interesting that we noticed is that after the retinectomy, 30% of the eyes are regenerated with multiple lens and multiple retina. So it will be really interesting to compare this retinectomy with the full amputation to understand which differences in pathways in signaling can actually have the control of the number of lens and the proper patterning of the tissue. And so in conclusion, today I show you how during my postdoc, I established a new research organism with camera eyes that are able to completely regenerate after full amputation, that has a direct embryonic development where we can do different amputation paradigms and we can do genome manipulation. And this will allow us really to try to understand mechanistically how the eyes can be regenerated and which is the redeployment of development programs. And with this, I would like to thank all the people that helped me out in this way and in this project. Alejandro is a really supportive mentor, and he really helped me out all the way in trying to establish a new system. All the lab for the great support and talk and bunch of ideas. The core facilities were really supportive during many steps of the experiments and our funding agency. Thank you so much. And I'm happy to take any question.
Great, thank you. It was really an amazing talk. We have already some questions, so we'll start with the Q&A. So we have the first question from Chris Wright. Is the early embryo, when you started to inject, mosaic or highly regulative? For example, at 4 cell stage or even a bit later, are the cells equipotent or not?
Yeah, that's a very good question. We inject them always at 1 cell stage, but we actually observe mosaic animals in F0. So we always want to have like a F1 and F2 before concluding any phenotype.
That makes sense. Another question also from Chris Wright. Work from others indicates the requirement for nerves in regeneration. Can you remove the optic nerve and ask the same question that you are asking?
That's a very great question. That's actually something that we are looking into. How much is the input and the requirement of having the optic nerve or not for regenerating the eye. So work in progress.
Another question from Jessica Feldman. Very cool talk. Any idea when during the process of regeneration, the eye/brain can perceive the light?
Yeah, that's also a really good question. We tried some behavioral experiments that were not really conclusive from that point of view. What we observed is with electron microscopy, looking at the ultrastructure of the photoreceptor, the fact that the photoreceptors are usually filled with vesicles that are good for like replacing opsins on the apical side, but also doing synapses on the basal side of the cell. And so we need 2.5 months to 3 months to actually reestablish the presence of these vesicles. So it's kind of an indirect evidence, but can maybe tell us something also about the function.
Great. Another question from Lila Solnika-Kretzel. Can other tissue or organs also fully regenerate in this model?
Yeah, very interesting. So they can regenerate the tentacles that you can see here. They have both a cephalic, really long tentacles close to the eyes, and the oral tentacles. I try to cut both of them, and both of them can regenerate. They can regenerate part of the shell. They can regenerate part of the foot. I never tried the organs inside the shell, but it would be really interesting to try.
Yeah, definitely. It is really, really cool. Another question from Morgan Goulding. Whoa, really nice work. Have you tried amputating other parts, so tentacles, section? And yeah, she just mentioned that she just asked a question actually. So perfect. Another question also from Chris Wright. For later response, okay, it's another question later. I had a question though. It's really broad and really naive. Do you know if there is any big gene duplication that happen in this species or not? Like, for example, we know in fish, we have this huge gene duplication. So gene A have multiple versions of the gene all around the genome. So do you know if there is any type of duplication? So for example, when you did your mutant on PAX6, is there?
Yeah, thank you for asking. Yeah, there is no a big duplication that we noticed, knocking out PAX6, and then we have also a couple of other lines that we obtain as mutants. We didn't notice anything major, but there is actually multiple, there are like, I think a total 6 genes that are annotated as PAX6. So the one that we targeted is because through bulk RNA-seq, we knew that it was highly expressed in the eye, and so that's why we picked that one. But there is another PAX6 that seems to be at least partially duplicated. So it's also possible that there are minor duplication, but not full genome.
I see. And are the others potentially contributing the mutants? So are they like more expressed when you have your PAX6 mutant, or did you ever check that already?
I tried, so with this other PAX6 that I tried to knock out, the animals have eyes and they are just behaving, they look like perfectly normal. So looking at the bulk RNA-seq combined with the phenotypes that we observed, I would say this is probably the PAX6.
I see. Cool, great, thank you. Another question from Crystal Rogers. So super exciting work. Do you have any idea why the animals develop duplicate lenses after removal of the retinas? Is there an increase in lens-specific gene expression in response to injury?
Yeah, so I don't have a full answer because I think it's an observation that I did during another experiment, and it's definitely something that I want to follow up. My hypothesis is that there is probably some signaling that is maybe not turned off, and because you can always see there is like the big lens, and this is consistent on the animals that we checked. There is a big lens more posteriorly, but then there are like new smaller lenses close to the cornea. So I don't know if maybe the signaling that should tell the cornea to stop producing new lenses maybe is still active, or like maybe there is a feedback that is not coming in. And yeah.
I see. Then I had a question also. So did you, because you mentioned it's really beautiful, your amazing pictures of the development where they seem kind of transparent and you could do potentially live imaging. Would you be able, or did you ever try, this type of system, I mean fish, so talking about like Toll2, to try to make like transgene to have your PAX6 cells GFP or this type of construct? Did you ever try or want to try?
Thank you. Yeah, we are trying meganuclease right now. And yeah, we tried already to inject a few things, and we are trying to find the perfect mix. But for now, we are testing meganuclease. That seems to be the classic approach for invertebrates.
Mm-hmm. I see.
Yeah, also Toll2, it's definitely something that we will try if we don't have like good results with meganuclease.
Mm-hmm. I mean, if your CRISPR apparently is working, so it is also possible to create transgenic line with CRISPR, but yeah, it's maybe not that efficient. But yeah.
Yeah, we have all of them in line, and like one at a time, we will try all of them until we succeed with one.
Yeah, no, for sure. I mean, there's so many cool stuff to do. It's really exciting.
Thank you.
So I think we reached the time. So again, thank you so much, Dr. Alice Accorsi. Your talk was really amazing.
Thank you.
So now we're gonna switch to the second speaker and give the talk to Dr. Elle Roberson.
Thanks, Joey. So it's my pleasure now to introduce our next speaker, Dr. Jiakun Chen. Jiakun received his PhD from Washington University in the Solnicka-Kretzel lab, where he studied the role of calcium signaling and cadherins in early zebrafish embryogenesis. He also developed and optimized genome editing tools like CRISPR to use in his zebrafish experiments. Jiakun is now a postdoc at the Vollum Institute at Oregon Health and Science University, where he's co-mentored in both the Freeman and Munk labs. And as a postdoc, he's been really focused on astrocyte development and function in Drosophila, and he's also discovered and characterized astrocytes in zebrafish. This work was recently published in Nature Neuroscience. And today, Jiakun will present his work investigating astrocyte morphogenesis and function in vivo. Take it away, Jiakun.
Thank you for the kind introduction, Ellie. Let me share my screen. All right. I'd like to also thank the organizers for offering this great opportunity to present my work. So my research is focused on how astrocytes develop and function in vivo. So we know that neurons are not alone in the central nervous systems. Glia cells are a crucial component in the CNS that is important for health and disease. And there are three major glial cell types in the CNS, this including astrocytes, oligodendrocytes, and microglias. And particularly in our human brains, glial cells compose about half of the brain cells. And astrocytes are the most abundant glial subtypes in human brains. They play diverse and essential role during the CNS development and function. For instance, they can have close contact with synapse to regulate their development activities. And they can also couple with blood vessels to provide nutrient support and homeostasis. And more exciting recent data also indicates that astrocytes can directly participate in neural circuit and behavior controls. And it is so that all these diverse functional astrocytes, depending on their morphological complexity, for instance, showing you here is a green label astrocyte, they typically extend numerous of fine process to interacting with surrounding cells. But we still have a very best question, don't quite understand is how do they acquire their complicated morphology, and does the complicated morphology really important for brain health and disease? So to begin to address this question, we start with the fruit fly model system. Fly have a relatively simple but sophisticated enough nervous system allow one to ask very interesting biology questions. And there are powerful molecular genetic tool sets, and their relatively short generation time really enable one to perform a rapid large-scale genetic screenings. In the fly brains, astrocyte also morphologically complicated. For instance, showing you here is a single astrocyte. You can see this fibrous process elaborating in the CNS. And this process partially is also in close contact with the synapse, likely mediating their activity and support their functions. So now we have a powerful model system to study astrocyte. How can we break it down to find gene important for astrocyte development function? So the straightforward answer in fly is doing a genetic screening. Yang Kang, who is a postdoc in our laboratory, and his interest in glial phagocytosis, and had done a large-scale genetic screening, identify a list of genes when disrupted affecting glial phagocytosis. And we reason some of the genes might actually affecting astrocyte development. So one particular gene I'm going to talk about today is a gene called TREE1. TREE1 is highly enriched in the fly astrocyte that is comparable to the traditional astrocyte enriched marker genes. So does TREE1 play a role in astrocyte development? So to test that, we first using an ionized strategy to knocking down TREE1. So here you are looking in the adult brain, antennal lobes, and the green is labeled the astrocyte membrane. In control, you can see the astrocytic fine process is densely infiltrated the entire field. That is make sure that the entire area is covered by the astrocyte membrane. However, if you have TREE1 RNAi construct expressing to disrupting this protein, this will resulting in more secret process of the astrocyte membrane. and they will not be able to fully elaborate, and oftentimes resulting in this gap region of the brain is not fully covered by the astrocyte membrane. And this data indicates that TREE1 likely play an important role to mediate the proper morphogenesis of astrocytes. So what is TREE1? TREE1 is actually encoding a lipid binding G protein coupled receptor, or GPCR. It is initially identified by Lutz Demonstrat in fly for its role in germ cell migration. And as a GPCR, there's two functional motifs. The NR1 or MPI1 motif is critical to mediate downstream signaling. And TREE1 is thought to be activated by still uncharacterized phospholipid ligands. So we decided to further explore TREE1 biology in astrocyte by examining the genetic mutants. So here we are looking again in the adult antennal lobes. In the control, you can see the green label astrocyte membrane is densely infiltrate the entire synaptic enriched neuropil to make sure the coverage of individual synapses. While in the TREE1 mutant, I hope you can appreciate the astrocyte structure is simplified, and oftentimes will result in gap region of neuropil not completely covered by the astrocyte membrane, similar to the RNAi phenotype I just showed to you. And this is a phenotype we can readily detect across different brain regions. For instance, we're looking at right now in the mushroom body regions. In control, the dashed line is labeled in the mushroom body lobes. You can see astrocyte membrane covering this area. However, in TREE1 mutant, you can immediately see that the deboil astrocytic process in this lobe region. You can also appreciate the neighboring brain region have also astrocyte morphology defects, indicating that TREE1 is indeed required for proper astrocyte development. And we're wondering whether this is also maybe a transient phenotype or persistent phenotype. So we're looking at larvae stage and also up to 30-day flies, and we can readily detect it in the TREE1 mutant. The astrocytic morphogenesis defects still persist at 1 month of flies, indicating when you lost this gene, this phenotype is irreversible. And so to further study the extent how loss of TREE1 could contribute to the astrocyte morphogenesis defect, we adapt a single cell strategy in which we can have individual astrocyte labeled by membrane GFP either in the control or in the TREE1 mutant background. And in control, you can see wild type astrocyte typically have this elaborate complicated morphology, while in the TREE1 mutant background, single astrocyte morphology is disrupted. And if we quantify the relative astrocyte volume, we can see individual astrocyte volume is significantly reduced. Next, we are wondering how astrocyte might affecting also cell-cell interaction. In this context, more like about astrocyte interaction. So to test that, we use a different system, a flip-out system, in which we can have all astrocyte labeled in green. And on top of that, we can flip single astrocyte from green to red. And you can accompany RNAi strategy in the red cell layer where we be able to study how wild type RNAi astrocyte interact with each other. So in the control scenario, if we simply just convert single astrocyte from green to red without any RNAi, and both on the green and red are wild type astrocyte, you can see they are densely elaborate in the neuropil region and forming their own territory that is minimally overlapping with each other. However, if we have the red cell also expressing TREE1 RNAi construct, you can see knocking down TREE1 will dramatically disrupt astrocyte morphology in the red clones. And interestingly, you can see that gap region in this red clone region now have been evaded by the neighboring white type green astrocytes, suggesting some interesting astrocyte tiling behavior being break down when you lost TREE1 genes. And this is something we are also working on. But we right now just simply focus on the red label astrocytes in control of TREE1 RNAi condition. We found that TREE1 knocking down in this single astrocyte can dramatically reduce its volume, similar to a mutant condition. And this data indicates that TREE1 is important for astrocyte morphogenesis function cell autonomously. And we are also interested in further exploring how the potentially downstream signaling and how TREE1 involved in astrocyte morphogenesis. So I'm going to summarize some additional experiments we have done. So using a series of rescuing construct like harboring mutation on those two functional motifs, and we can specifically drive their expression in astrocyte. And we found that the first motif is dispensable, while the second MPR motif is critically important for astrocyte function in by TREE1. And this is slightly different from what people have found for TREE1 in germ cells. In addition, we found that TREE1 can genetically interact with small GD-page RED1 to regulate actin cytoskeleton to ensure morphological complexity of actual size. And one piece of data I'm going to show you today is that if we use a lab-acting GLP marker to label the actin cytoskeleton or individual actual site, which will allow us to reconstruct the 3D branching pattern of individual actual site, we found that in control, you can see the complicated branching pattern of this actual site. However, in TREE1 background, oftentimes the actin cytoskeleton structure is dramatically simplified. And we quantify the total filament length adding up together, mutant actual site is dramatically reduced. And this data is interesting, suggesting that TREE1 regulate actual site morphogenesis likely mediate through the actin cytoskeleton rearrangements. And we are wondering whether we can directly visualize or monitor how loss of TREE1 gene resulting in this phenotype. And fly is good for as a genetic models, but it remains challenging to directly lab imaging adult brains. So we decided to turn to a different model organisms. So we wondering zebrafish could be a well-suited model system to study this, because zebrafish have been used to study cellular behavior and cell-cell interactions by time-lapse imaging approaches. And the first obvious question we have to ask whether TREE1 GPCR function is conserved in vertebrate actual size. And although glia is an ancient cell type that is conserved from simple mobile organisms such as C. elegans and fruit fly to humans, it has been remained controversial for a while whether zebrafish do have true actual size. And part of the reason is owing to the continuous regeneration capacity in this species. And when I start my postdoctoral, I'm also reasoning whether it is potentially owing to a lack of genetic tool to carefully characterize actual size in zebrafish. So by generating a new set of genetic and molecular tools, we recently report that zebrafish do have actual size, and they are morphologically and functionally quite equivalent to their counterparts in other model systems. And particularly using live imaging, we can actually monitor a single actual site derived from their progenitor cell. In this case, you're seeing early stage, this is more resembling a radioglia morphological progenitor cell that have a long process extending from soma. And across time, you can see that elaborating dynamic process, and eventually we are forming a morphologically complicated actual size. And because this is all imaging in live, we can actually turn back to or monitor the track the same cell across several different days. And using this strategy, we actually define the time window critical for actual site morphogenesis to occur in the zebrafish larvae CNS. And this is roughly between day 2 to day 4 post-fertilization. They will rapidly growing in size to maintain and their related territory outer walls. And now we have a powerful system to also study actual site, particularly from the live imaging perspective. We decide to turn back to a question, what is the likely equivalent of Drosophila TREE1 may be involved in vertebrate actual site development? So the first obvious candidate is the closely related GPCR hormonal TREE1. So in vertebrate, it's a gene called GPR84. And so we generate mutant for GPR84 and looking at the data stage at day 6 post-fertilization when actual site morphology is relatively established. And here you are looking in the spinal cord compared to their control sibling versus homozygous mutant. We find that loss of this gene did not dramatically or affect the actual site morphology at all, indicating GPR84 is likely not the vertebrate GPCR involved in this process. So with that, we decided to further exploring additional GPCR. We reasoned that likely still would be a lipid binding GPCR. And we found one particular one, S1PR1, short for sphingosine-1 phosphate receptor 1, is highly enriched in the mammalian actual site populations. And using in situ hybridization experiment, we found in zebrafish larvae S1PR1 is also highly enriched in the brain and also in the spinal cord. So to test whether S1PR1 play a role in actual site development, we generate 2 independent alleles of the S1PR1 genes using CRISPR-Cas9 assays. So using these mutants, we now can come back to see whether they have phenotype, and they do have phenotype. And again, this is looking at later stage at day 6 post-fertilization, actual site morphology is relatively established. In the lab panel is showing you in the spinal cord, and the actual site membrane labeled by green is densely typically densely infiltrate the two lateral neural pair enriched region to covering the entire neural pair. However, if you lose of this receptor, this will result in the disruption of this associated elaboration in the lateral neural pair enriched region. And this phenotype can be observed also similar to tree 1 in flight across different CNS regions. In the right panel is showing you different brain region, and the dashed line is labeled where typically in wild type the associated process densely infiltrated. If you have the S1PR1 deleted, this density infiltration phenotype is also dramatically disrupted, indicating that S1PR1 is likely the vertebrate GPCR involved in astrocyte development. And so now we know S1PR1 playing a role in astrocyte development in zebrafish. We turn back to the early time point when the time point astrocyte is still actively growing, which is between day 2 to day 4 post-fertilization. And using mosaic labeling, we can actually monitor individual astrocyte fine process growing. And you can see from the movie, both control and mutant astrocyte, they will have fine process extending and retracting in the temporal manners. But oftentimes we find that the fine process in mutant case seem to have a faster turnover rate. And it's probably easier to see in this still time-lapse image. Usually wild type fine process is more persistent, while in mutant, sometimes we can even see the reconfiguration of their rearrangement from frame to frame. And we quantify the relative extension and retraction speed of individual associated fine process in wild type versus mutant. We see both the extension and retraction speed is faster when you have the receptor deleted. So this data in conclusion suggesting that S1PR1 might play an important role to regulate the proper dynamics of associated fine process to ensure they can forming a complicated morphology. And the last question we are really interested in is whether this receptor is required throughout or maybe just during the active growing phase. As I mentioned, the day 2 to day 4 period, they are active growing in size. So to test that, we can use specific inhibitor to block the receptor signaling, and that's 2 different drugs we apply. So we can inhibit receptor signaling in a temporal manner, either during the active growing phase, which is between day 2 to day 4 post-fertilizations, and then see what happens at day 6. Or we can allow them to grow until day 4 post-fertilization and start to blocking the receptor signaling and see what happened to the astrocyte morphologies. And using this strategy, we first looking at the early treatment time points when the astrocyte is active growing and we block the receptor signaling. And you can see both drugs can dramatically disrupting the associated process typically we see in wild type, this dense GFP signal in the neural pair region. And this is also quantified by the GFP signal in this dense area that are dramatically reduced when you have the receptor signal being blocked. And surprisingly or interestingly, if we allow them to develop until day 4 post-fertilization and start to blocking the receptor signaling, we almost see an exact similar phenotype as early treatment. So later blocking of this receptor can also comparably disrupting already established morphology, meaning that the receptor signal is not simply required during this active growth phase, but also during this later growth phase of astrocyte morphogenesis to be established. So with that, so I'm showing you there's likely conserved GPCR signal mediated by TREE1 and S1PR1 is important for astrocyte morphogenesis from fruit fly to zebrafish. And we are also studying the potential upstream phospholipid ligand involved in this process and how the ligand processing enzyme UNAN or LPP might play a role in astrocyte development. And for future, one interesting direction we are really actively pursuing is how does disrupting astrocyte morphology, going back to my introduction slide, whether that's important for neural circuit assembly and functions. So to address these questions, we can actually take advantage of each model system to slightly answer the question differently. So for instance, in fly, there have been defined circuits that have been well characterized by previous studies. And we can easily use our mutant or specific disrupting the genes we identify in astrocyte and see how does that affect, is that the neural circuit assembly or their maintenance. While in zebrafish using live imaging, particularly using calcium imaging, we can study how astrocyte might regulating or modulating neural activity, or when astrocyte morphology is disrupted, how does that affect the extracellular neuron communications. So with that, I would like to thank all the people in the Mount and Freeman labs, and particularly my mentor, Kelly and Mark, for their full support. And I would also like to particularly thank Cam, Youngsik, Taylor, and Toby for their support with this project. And thank you for your attention. I'm happy to take questions.
Jiakun, that was a really wonderful talk. You have many questions coming in. So the first question is from Zainab Afzal, and they ask, I was curious about the green wild type and red TRE1 mutant astrocytes you showed. When the green ones are invading the red area, are the red ones dying, or is it the green ones have increased growth or are just invading?
So that's an interesting question. So astrocyte is thought to be tiring with each other to make sure the entire CNS is fully covered. So in this case, the red one is not dying, but the morphology of that one is disrupted because those of the TRE1 receptor. But somehow in this case, the white type or the green one start to infiltrate because they saw all their space they need to be taking up to compensate for the loss of the other cells.
Great. Jessica Feldman asks, given the highly branched cell morphology in these astrocytes, do you know what mode of actin polymerization they are using? ARP2-3 branch network versus a more filopodial network or a combination?
Yeah, that's an interesting question. We haven't fully explored in this direction, but we have some data pointing to the small GTPase RED1, and one likely mechanism is that the receptor may be recruiting RED1 to promote the filopodial protrusion or stabilizations.
Great. And so Chris Wright is also asking a question. He says, sorry if I missed this, Jiakun, but are there any human GWAS or other studies indicating problems in this gene?
Yeah, so this gene actually has been found to be involved in autoimmune brain disease. And also the drug that I'm used to actually treat the fish to blocking the receptor, the fingerling actually is being active use in MS patient it is thought to prevent the immune cell to invade the brain, but we think maybe that's also a mechanism in astrocyte. That's something that we are also interested in.
Cool. And finally, we have a question from Michelle Stars Guyano. They say, beautiful work, Jiakun. Can you rescue the TRE1 mutant phenotypes with activated RAC or similar actin regulators?
Yeah, that's an interesting question. So we actually have done experiment. We use either express a constant active RAC or we express a dominant negative RAC in the TRE1 mutant background or in wild type background. But we find that oftentimes either inhibiting or activating RAC activity will cause a phenotype itself. So we reason that likely receptor is more like balancing the RAC activity, but not simply going for one direction to mediate this process.
Oh, interesting. I also had just a couple of questions. So I was really interested by that phospholipid gradient that you said activates TRE1 and potentially S1PR1. And I was wondering if you could tell us a little bit more about that, like where would that gradient necessarily come from, or if even the membrane of astrocytes are composed of different types of lipid moieties compared to other neurons?
Yeah, so this is an interesting question. So in vertebrate, the S1PR receptor is thought to be activated by the sphingosine-1 phosphate. And there's enzyme involved in phosphorylate the sphingosine to become S1P. And some preliminary data I've done shows that in vertebrate or in fish, that's the case. This is likely the enzyme involved in generating the ligand, but we don't know exactly where it's coming from, either from astrocyte or neurons. That's something that we are also working on.
Cool, great, thank you. Marsha Lucas asks, I may have missed this, but do the S1PR1 zebrafish mutants grow to adults?
Yeah, so they do grow to adulthood. We are collaborating with some behavioral groups to figure out whether we can find an assay to see whether in the larvae stage there's some phenotype. But even say in flies, the TRE1 mutant can grow to adulthood. But interestingly, we find they do have a short lifespan, and particularly when they're aging, there's a locomotion defects, and so that's clear something going on when you disrupt astrocyte morphology, but that's more for experiment to be explored for sure.
Great, thank you. Okay, I think that I will thank both of the speakers that we had today. So thank you so much, Alice and Jiakun. You guys both gave really amazing, excellent talks. Just as a reminder, this seminar has been recorded and it's going to be available on the SDB website starting on Monday. So please join us next month. The next month's seminar is on Friday, April 8th, and we'll hear from Joshua Abrams from New York University School of Medicine at the Skirball Institute, as well as Catherine Criswell from the University of Cambridge. Thank you all for coming.